back and look at all the awards you've won, because too many to just remember.
Aw. Thanks.
It's okay to brag about you a little bit.
Oh my gosh, I tried to restart my computer this morning and it wasn't working. It took like 15 minutes and then we just like turned it off and turned it back on, and I was getting really nervous.
Nothing like a little bit of stress to energize you for your talk. So
right?
Consider it a good thing.
Hopefully.
Okay, it looks like people are joining here. So, well, I'm gonna go ahead and get going. So, well, welcome everybody to the Master's Defense for Christi Kruse. I'm so happy to be introducing her right here, because it's been just a wonderful time working with her in the lab and spending time in the field together. And for those of you familiar with the lab group, you know that we prefer to work in desert ecosystems. We have a lot of projects down in Arizona, Southern California, Northern Mexico. It's just a place that's interesting to us. And in fact, I met Christi at the Desert Fishes Council meetings in Furnace Creek down in Death Valley, California. And we first talked about the possibility of graduate school then. And at the time, she was working with California Department of Fish and Wildlife out of Bishop, California, on fish populations and desert ecosystems. And so it seemed like a natural fit that Christi would come. We'd work on deserts together, and she'd get her master's degree here out of OSU with me. Well, and I think this is a testament to Christi's putting questions ahead of systems. When we really dug down and thought about the question that would be interesting to tackle, it turned out that the best place to study it was in Oregon. So, Christi, I commend you for sticking through the Oregon winters to get the best data possible. And just a quick note on her research. Within the realm of river ecology and stream ecology, there's a paradigm known as the natural flow regime. That's a unifying principle for how we understand the ecology and management and even the evolution of river ecosystems. But there's not a similar framework for wetlands and ponds that dry up seasonally. And so Christi's research is start in that direction. So this is the beginnings of the natural hydroperiod regime and the importance for the organization of ecological systems in those kinds of habitats. So it's really original research in that sense. Just a few things about Christi. She got her undergraduate degree at George Mason University. She's from Virginia originally. Then went out to Bishop, California and became a desert rat. And I think you're probably going to turn back into one after this master's is done too. And during her time here, she's been really successful getting awards. From the Society for Freshwater Science, she received both the Bosel-Sanderson Award for Natural History, the Sheldon Award for Population and Community Ecology, and the society actually had to cut her off from winning more awards because she's just too good at it. And since then, she just finished a U.S. Fish and Wildlife Service Directorate Fellowship, which is a prestigious fellowship awarded by that service that then gives them direct hire authority to hire her into a permanent position anywhere in the nation. So it's a really great fellowship to get and a good career launcher for post-master's degree life. So in addition to that, I know Christi is also a talented visual artist, and I hear that she's really good at stick and poke tattoos. So you can talk to her about that directly. So alternative career choices there are possible. So in any case, no further ado, Christi, take it away.
Thank you, Dave. That's a good intro. All right. All right. Does this look good?
That looks right on.
Okay, cool. All right, great. Okay. Hi, everyone. Thank you for joining for my presentation. This should take about 42 minutes. And then there should be some time for questions at the end. For this presentation, I'm first going to go through my research studying temporary ponds, where I'll discuss the context and background of my work, my research questions and predictions, the methods that I use to test my predictions, the results of the study, and then finally my conclusion. And then I'm going to discuss my experience with disability as a graduate student and some suggestions that I have for making science and academia more accessible and inclusive for people with disabilities. At the bottom of the screen, you'll be able to see a little roadmap of my talk, and I will be highlighting the section that I'm discussing on each slide. So as Dave mentioned, I'm part of a research group at OSU that studies the natural patterns of flow in a river, such as flood or drought, and how those patterns influence the invertebrates that live there. So in addition to invertebrates, the whole plant and animal community living in a given river, known as the biotic community, will be adapted to natural patterns of flow that occur in that river. Thus, the natural patterns of flow are considered to be the primary factors that organize and define biotic communities of a river. The patterns of flow can be defined in terms of the timing, duration, magnitude, frequency, and rate of change of those flow events. The natural flow regime explains biotic communities of rivers well, but in ecology, the most useful concepts are those that can be applied across many ecosystems or many habitat types. So to see if this idea is transferable, I'm applying the parameter of duration to ponds. I want to know how the duration of time that a pond holds water influences the invertebrates that live there. The duration of time that a pond holds water is known as the hydroperiod, and this is going to be a concept that I'll refer to throughout the talk. We can think of hydroperiod as a hypothetical gradient, and on one end of the spectrum, we can have a puddle, that lasts for about an hour. And then on the other end of the spectrum, we have the oldest lake in the world, which is 24 million years old. In between, we typically consider waters that persist for more than 1 year as perennial, and less than 1 year is temporary, although there can be some variation in these distinctions. In this study, I'll be focusing mainly on temporary waters with hydroperiods less than 1 year. These photos show a temporary pond while it was inundated with water and while it was dry. While the pond was holding water, it was habitat to aquatic invertebrates, but when it was dry, it was not a good habitat to those same animals. So to predict how variations in hydroperiod length may influence the invertebrates that live in a temporary pond community, we first have to understand how the invertebrates get there in the first place and where they go when the pond dries. To start, we're going to consider this hypothetical hydrograph of a temporary pond. We have time on the x-axis and depth on the y-axis. Once the pond starts to fill with water, over on the left side of the graph, it will get deeper and then hold a relatively constant depth until the pond starts to dry. We'll consider this portion of time on the left side of the hydrograph to be the filling phase. Aquatic invertebrates will start joining the community at the start of the filling phase, and they will join through flying or crawling into the habitat, or some invertebrates will actually be in a dormant phase in the pond substrate. And so when the water returns, those animals will reanimate and join the community. We're going to call this period of time the assembly phase. And phone's distracting me. Once the pond begins to dry, the habitat will start to shrink. And at this point, as the habitats are getting smaller, new colonization events by animals that are flying or crawling in will diminish because they're not as attracted to being in smaller habitats. And so this will be kind of the period of time when new colonizations will be diminishing. And so we're going to call this the invertebrate community assembly phase. While the pond is drying, there will also be a decline in the water depth, dissolved oxygen, increases in temperature, and the invertebrates will become more concentrated within the remaining water habitat. These shifts will cause the community to begin to disassemble. Invertebrates that are vulnerable to these environmental changes will leave the community sooner, and the resilient invertebrates will stay longer. When invertebrates leave, they will either emigrate by flying or crawling or burrowing out of the habitat. They'll go dormant back into the pond substrate, or they'll die. To address my research question, which asks, how does hydroperiod length influence invertebrate communities in temporary ponds? I studied ponds that represented a range of hydroperiod lengths. So we have that same hypothetical hydrograph, but rather than just one pond, we actually see 4 ponds here. And each starts at the same point in time and goes completely dry at different points in time. So I used the concepts of assembly and disassembly as frameworks for making predictions about how invertebrate communities would change in response to pond hydroperiod length. However, I also acknowledge that there are other processes that influence invertebrate communities, and assembly and disassembly will not account for all of the community variation among the ponds that I studied. I decided to use assembly and disassembly as frameworks for addressing my research question because they predict community changes based on different mechanisms. Assembly predicts that community variation will be the result of habitat duration and is a general concept that, that can be applied to many ecosystems, while disassembly predicts that communities will be fairly similar throughout the hydroperiod, and variation in the communities from the beginning to the end will be caused by environmental shifts. And this is specifically tailored to ephemeral or temporary ecosystems. So assembly, going back to assembly processes predict that the longer the hydroperiod or the longer the habitat duration, the more time there is for colonization events by invertebrates. So under this framework, I predict that ponds with longer hydroperiods will have more species turnover, which is a measure of how invertebrate communities change with time in regard to species composition and species abundance. Next, I predict that ponds with longer hydroperiods will have higher species richness, which is basically just a count of the number of different kinds of species in the community. And then finally, I predict that ponds with longer hydroperiod would have higher abundances of predators. For my disassembly predictions, I expected that the changing environmental conditions as ponds dried would cause changes in the invertebrate communities. So under this framework, we should see shifts in species traits between the filling and drying phase as the environmental conditions shift. We assumed that when ponds dried, the dissolved oxygen levels in the water would decrease. And so we expected to see a higher proportion of taxa with traits for breathing atmospheric air in the drying phase. Excuse me. And next, we also expect that as habitats shrink, the encounter rates of the invertebrates left in the pond, those encounter rates will increase, and the animals with protective body coverings would persist longer. So we expected to see a higher proportion of animals with protective body coverings. To conduct my research, I worked in Willamette Valley, Oregon, which is an ideal place to study temporary ponds because the region has rainy, cool winters and warm, dry summers. Historically, the valley was managed by the Kalapuya people, who maintained about 1/3 of the valley as seasonal wetlands. However, when European settlers arrived, they forcibly removed the Kalapuya people and cultivated or developed all but 2% of the historic wetlands. Recent conservation efforts have started to re-establish temporary waters, and so my research was conducted entirely in restored habitats. I collaborated with local landowners and land managers to locate temporary ponds that were rain-fed and did not have surface water connections to other perennial water bodies. I selected 9 ponds that I predicted would represent a range of hydroperiod lengths based on the surface area and depth of the ponds when they were dry. And so the ponds that I selected ended up representing hydroperiods that ranged from 146 to 215 days. You can see on the map that I have each pond located and the hydroperiod length of the pond labeled next to the point. I also studied one perennial pond as a point of comparison for invertebrate communities in a much longer hydroperiod system. I wanted to study a fishless perennial pond so that it would be more comparable to the temporary ponds because the temporary ponds are fishless and fish can have a dramatic effect on invertebrate communities. However, I couldn't find a perennial pond that was fishless, and so instead I settled on a perennial pond that was not actively stocked with fish. And that's marked up here as 365 plus days on the map. I installed staff gauges and pressure transducers in each pond to monitor pond depths and determine the filling and drying dates of the ponds. And so here I'm standing next to a staff gauge and the pressure transducer that actually fits down inside of the staff gauge. So it's also a stilling well. Let's see. I collected aquatic invertebrate samples from each pond 1 month after, once per month for the duration of the hydroperiod cycle of each pond. I collected samples by sweeping a 500 micrometer D-net across pond substrate, covering 6 meters squared per minute. I dispersed my collecting efforts across pond microhabitats, so the sample would be representative of all of the different kinds of invertebrates that lived in each pond community. I processed samples from each pond 1 month, within 1 month of when ponds started filling and within 1 month of when ponds started drying, so that I could have kind of the full range of the community development. And then invertebrates were identified to the lowest practical taxonomic grouping. Insects were mostly identified to genus, and non-insects were identified to various taxonomic resolutions because I have less expertise with working with non-insect invertebrates. For some of our analyses, I decided to test insect and non-insect taxa separately. And this is because I expected there to be different community trends between these two groups. Non-insect taxa are passive dispersers, meaning that they cannot fly, and they have a dormant life stage, meaning that they will be early colonizers of the pond, reanimating when the pond starts to fill, while insects have a lot more variation in their life history strategies. And so I expected that if I separated these two groups, I would be able to have a finer resolution on the community trends. To address my prediction related to community turnover, I wanted to evaluate changes in community composition and changes in taxa abundance. So I constructed a non-metric multi-dimensional scaling ordination. I used log transform total abundances of the taxa from each pond and the Bray-Curtis dissimilarity measure and to create this ordination. So to orient you to what we're looking at, each point represents one pond sample. Circles represent drying samples and triangles represent filling samples. The hydroperiod length of each pond is also labeled next to the sample so that you can figure out which sample you're looking at. And then filling and drying samples of each pond are connected by a line. Points on this ordination that are closer together indicate that those communities are more similar, and points that are farther away indicate that they're less similar in terms of the species composition and the abundance of those species. I also included the perennial pond in this ordination, which is in the upper right corner, of the ordination. So you can see that most ponds in the filling samples grouped pretty closely together, which means that these communities were fairly similar. And most ponds in the drying phase sample also grouped together, meaning that these communities were similar. The large separation between the filling and drying samples of the temporary ponds indicates that there was a large amount of community turnover that occurred between the sample collection points. You can see that there are also 2 temporary ponds that didn't follow the same trend, and I think that these are just an example of other factors that are influencing the invertebrate communities that I studied. So we were interested in seeing if longer hydro period meant that ponds would have more community turnover. And if we look at the distance between the temporary pond points, we don't necessarily see a trend that indicates that longer hydro periods have more community turnover or those points are farther apart. And so we did not see what we predicted in this analysis. However, we did find something interesting, which was that the temporary pond communities were very similar between phases, and they shifted from the filling to drying phase in similar manners. And they were also very different from the community composition, and the amount of turnover that we observed in the perennial pond. For the remainder of my hypotheses, I'm going to test my predictions in two ways. First, I'm going to compare the correlation between hydroperiod length and the community trait of interest. So in this case, I'm looking at taxonomic richness. And this method is used to confirm the association between whatever community trait I'm looking for and the hydroperiod length. So do we see variation as hydroperiod length increases? I'm also going to test my predictions using a box plot, and this method will illustrate if pond communities shift without considering the variation based on hydroperiod length. And so in this sense, we're just taking the mean value of all of the different ponds in the filling phase and comparing them to the mean value from all the different ponds in the drying phase. Okay, so those are the two methods I'm going to use, and they'll pop up throughout the rest of this analysis section. So looking at taxonomic richness, I expected that taxonomic richness of ponds would increase with pond hydroperiod length. However, I did not observe that in my insect or my non-insect communities. However, I did observe that for insect taxonomic richness, there was a significant increase between the filling and drying phase, which we did not observe in our non-insect taxa. Next, we wanted to test the prediction that predatory abundance would be higher in longer hydroperiod ponds, and we also did not observe that prediction. However, we did observe that non-insect predators had a significant decrease in abundance as ponds shifted from the filling to drying phase, and that was also not what we expected, but a pretty interesting result. We didn't see what we expected to see under our assembly predictions. However, I found something interesting when I looked at the hydrographs produced by the pressure transducers. So this is a hydrograph produced by one of the ponds. And you can see that the pond started to fill mid-December and then transitioned to the filling phase on April 23rd. So this was actually similar between all of the ponds that I studied, except for one, that they started filling on the same date and they transitioned to the drying phase on the same date or around the same date. And so this tells us that the filling phase length of most of the temporary ponds that I studied they were actually very similar, even though the ponds had different total hydroperiod lengths. Now we know that the majority of community assembly occurs during the filling phase. And so I think that this trend actually may explain why we didn't see more variation in the pond communities based on our assembly predictions, because most ponds likely stayed in the filling phase for similar lengths of time, or stayed in the assembly phase for similar lengths of time. Next, we're going to move into the disassembly predictions. So I predicted that the proportional abundance of taxa that breathe air would increase between the filling to drying phase. And we did not observe that prediction. However, we did actually finally see a correlation between a trait and hydroperiod length. So we observed that in the filling and drying phases, there were correlations where the proportional abundance of invertebrates that breathe air increase as the hydroperiod length of that pond increases. However, when I looked at the actual taxa that were identified under this trait, the majority of them were gastropods or snails. And so it was 3 different families of snails and then leeches. And so I actually think that this is a characteristic more related to snail biology than it is actually for this trait as an indicator of community disassembly. And then finally, we predicted that the proportional abundance of animals with protective body coverings would be higher in the drying phase as compared to the filling phase. And so to do this analysis, we split our taxa, our non-insects into, or our insect and non-insect taxa into full armor, partial armor, and no armor, just depending on how much of their body was covered by protective coverings. And we did finally see the results that we expected for our non-insect taxa only. There was not a significant shift with full armor, but for partial armor, there was an increase, and for no armor, there was a decrease. And so this actually did finally follow the predictions that we were expecting. So overall, we didn't necessarily observe what we expected to see, but our results gave us some good insights into how hydroperiod influences invertebrate communities. So first, invertebrate communities were similar among temporary ponds, which is not what we expected, but it's likely related to the similarities in the filling phase lengths between our temporary ponds. Next, community trends were very different between our insect and non-insect taxa, which was really interesting. And I think that this is due to differences in life history strategies of our insect versus non-insect taxa. And our insect taxa were actually more coarsely grouped. There were 3 life history strategies included within the insect taxa. And so I didn't have enough information about each animal to split them into their life history groups, but I think in the future, this could be a really interesting extra level of resolution to look at community trends through. And then finally, we concluded that hydroperiods less than 1 year actually seemed to be more of a unifying influence on the invertebrate communities than the variation in hydroperiod lengths between the temporary ponds. And so this seems to agree somewhat with our disassembly framework that predicted that communities in temporary ponds within a landscape would be similar to each other. Although we also only tested hydroperiods that ranged from 146 to 215 days, I believe. And so it's possible that if we tested a wider gradient, so maybe like 30 days to 300 days, we would see more variation. But overall, we conclude that hydroperiod length is influential in invertebrate communities in ponds, just as the duration of flow events are influential in rivers. And so this does give some merit to the idea of expanding the concept of the natural flow regime to other ecosystems. And I think that this would be could be an informative way to look at other ecosystems or other ponds with a larger range of hydroperiod lengths. Okay, I'm gonna take some water. So now I'm going to transition into the second part of my talk. Typically, defense presentations just pertain to research, but in my case, I'm choosing to share a reflection of my experience with disability as it pertains to academia, with the hope that sharing my thoughts will lead to more space for students with disabilities to participate in science in the future. So, I also made all the art in this section. Okay, so to start, I'd like to define disability. The legal definition of disability under the Americans with Disability Act is a physical or mental impairment that substantially limits one or more activities. However, during this talk, I would like to paint a picture of disability as a social construct. So rather than thinking of disability, as something wrong with a person, I would like to define disability as variation in human function that is not accommodated by society. So during this talk, I'm also going to refer to the concept of an invisible disability, which is a disability that goes largely unnoticed. In my case, the disability that I have is that I read more slowly than my peers. It is classified as a learning disability because within higher education, academic institutions, students are expected to be able to read at a particular pace. On top of my learning disability, in 2018, when I arrived at OSU, I was in the early stages of recovery from a traumatic brain injury. The injury caused impairments in my sleep, focus, and visual processing. And in school, this meant that it was difficult for me to focus on conversations or tasks because I was easily distracted by visual or auditory stimulus, which made it difficult for me to commit things to memory. And also it caused me to work more slowly. At this point in time, right now, my brain injury is almost completely healed, but my condition caused me to experience academia through a more dramatic lens of disability during the first 2 years of my graduate program. Before I had my brain injury, I already had a pretty negative view of my own learning disability, which I developed in response to my experiences in higher education. So I've always read more slowly than my peers, but during grade school, reading slow just meant that I took longer to read, and it didn't actually prevent me from being a high achieving student. My slow reading speed became a significant problem when I was preparing to apply to college. I couldn't read fast enough to complete the SAT, which is a timed entrance exam, and so my scores were too low to be useful for me. My family paid $1,500 for me to have a learning evaluation done, which concluded that I had a reading disorder. The formal documentation provided by the evaluation allowed me to take the SAT with extended time, and I was admitted to George Mason University in Virginia. My diagnosis gave me access to my textbooks on tape and screen readers, which helped me to complete my work more efficiently, which was great, but my new diagnosis also exposed me to the social stigma around disability for the first time. So prior to my diagnosis, my peers and teachers in high school regarded me as bright and capable, which was how I saw myself. But after my diagnosis, when I would share my new identity with my peers and professors, I started getting negative comments. People would call me slow, which I think was implying that I was stupid. And they would also project my learning disability onto other cognitive functions that were not impaired. Excuse me. So people were creating inaccurate narratives about me. These experiences taught me to stop sharing my identity freely because people seemed to see and judge me more favorably if they didn't know that I had a learning disability. However, this mistreatment made me feel ashamed of myself as I was leaving college. And it kind of just, the negativity stayed with me. So the challenge with reading in these confined contexts continued when I was ready to apply to grad school. At this point, my educational evaluation had expired and I couldn't afford a new evaluation because I was working seasonally. And so I took the GRE without extended time, and predictably my scores were too low to be useful. I tried to figure out other ways to progress my career without the GRE, but it just wasn't working. These high quality graduate programs at the time, so this was like 2012 when I was ready to apply, these high quality graduate programs all required the GRE. And so I ultimately asked my family to pay for another educational evaluation, which granted me extended time and I got high scores. And so ultimately after 6 years, I was admitted to OSU. And so I was really excited about being admitted, but the experience that I had with the GRE also made me wonder if I actually had the capacity to succeed in grad school, or if this test was accurately keeping me out of grad school, because these tests are in theory used to predict who can be successful in a graduate program. So when I arrived at OSU, I had a lot of doubts because of my perception of my learning disability and difficulties caused by my brain injuries, but I had a couple of really big benefits. During the first year of grad school, my advisor gave me an office all by myself because of my brain injury, and that was hugely helpful in helping me manage my distractions and kind of get my new learning strategies together. And then the second advantage that I had is because I've always learned differently than the majority of my peers. I've always had to apply extra determination and creativity to my work. And so when I needed to develop my new adaptive learning strategies to work around my challenges with my brain injury, it was pretty intuitive for me. So these are the new strategies that I developed. I started using sound canceling headphones. I took notes on almost everything, especially conversations. I used voice recordings of myself so that I didn't just lose my thoughts all the time. And then I used text-to-speech screen readers. And I actually found that there is an excellent resource available on all Microsoft computers. If you open a PDF in the Microsoft Edge web browser, there is a button that says read aloud. So you just highlight the text that you'd like to have read aloud and it will read your PDFs to you. And it's actually really high quality. It sounds like someone's just talking to you rather than a robot reading to you. So if anyone's interested in that, that's there. I would also use annotation tools while I was reading, so I would highlight and take notes to keep myself focused. And I use hourly planners to actually track and cue myself on what I should be doing at each hour of the day. So these strategies worked really well for me. I got A's in my classes, and as Dave mentioned, I've received a handful of awards over the time in school, but the difficulties that I had with my deficiencies were still pretty difficult for me in social contexts of being a graduate student. So in addition to my classes and research, which were things that I was handling really well, I was also obligated to teach, to participate in teacher trainings, to go to lab meetings, department meetings, and graduate committee meetings. And these group settings would be difficult for me because sometimes we would be asked to read hard copy text. So this would be usually a teaching technique for workshops or peer revision groups. We were handed a piece of paper with text on it. We were given an allotted amount of time to sit there and read it, and then we would be expected to share our response with the group. This environment was really difficult for me because at the time I was primarily reading with electronic text screen readers. And so reading hard copy text was just more difficult. It was difficult for me to focus, and I was a little bit more slow in these contexts. I knew that I read more slowly than my peers, so I felt anxious. And then, yeah, like with the distractions, there were people around, there were lights, I was nervous, there was just a lot going on. And so in these contexts, I usually didn't actually have time to comprehend the text at the time when it was, when I was time to share with the group. And so I would always feel put on the spot to share my thoughts. I had the option to advocate for myself in some of these situations when I knew what I was walking into. I could ask to have the text in electronic format, or I could ask for extended time. And even though I knew that that advocating for myself would help me participate, I actually felt more comfortable not advocating for myself and just pretending like I could handle the assignment along with everyone else. So this is important. I declined I decided not to advocate for myself because I risked opening myself up to the similar mistreatment that I experienced as an undergrad student. And I felt this way. I didn't see any cues that would have indicated to me that I would have a different experience because within my graduate department, I never heard disability being discussed amongst my peers or the professors that I worked with. I didn't know any other students or professors in my graduate department who needed accommodations for disability. And it seemed like because my disability was not visual, everyone assumed that it was not there and that I could read quickly. And so I also had this level of assumption of, who I was that wasn't true. And so rather than advocating for myself, I just tried to fit in, but by trying to fit in, I just felt more uncomfortable. My negative view of my own disability started to change once I had my first opportunity to mentor. It was the spring of my first year, and I started working with an undergraduate student named Megan. I identified with her because she was also dealing with challenges related to a brain condition. Megan helped me conduct my research by collecting invertebrate samples in the field and then helping me process those samples in the lab. I also mentored her on an independent research project where she studied a beetle called Apterolipus parvulus. It's right here, and it's about the size of a sesame seed. So this beetle is flightless, and it was prevalent in a lot of my field sites, but really interestingly, it had only been identified in a couple of locations outside of the Willamette Valley. Excuse me. We were really curious about this beetle because it's flightless and it lives in temporary ponds. So we were wondering, where does this beetle go when the pond dries? Megan conducted a field study to start to address this question. And then we also looked at data from samples that I collected. And together we were able to describe the life cycle of this beetle. And we're still working on this project, but we're hoping to publish her work in a natural history journal in the future with Megan as the first author. So really cool stuff that we were doing. But while we were working together, Megan would also describe to me the pride that she held in her identity as having an invisible disability, which is due to a medical condition. She described tools that she's used to navigate an education system that does not accommodate her needs and strategies that she's used to advocate for herself and others. She also described something to me that she calls superpowers, So having a disability is actually a trade-off. A person with a disability is faced with more challenges than a person who does not have that disability would experience. And navigating these challenges requires creative problem solving and developing skill sets that are not required by the general population. And so in result, people with disabilities have a disability, but they also have some pretty extraordinary strengths. So rather than distancing herself from her disability, as I had been trying to do, Megan was embracing it and seeing the strength in herself. This was the first time that I had heard of disability being reframed in a positive light, and I was inspired by her self-compassion and the care that she gave to others with disabilities. Over the course of my graduate program, I connected with other undergraduate students with disabilities in various capacities. And like me, each was feeling the pressure of studying within an environment that was not designed for them. So I realized that by embracing my own identity as having a disability, I could actually use my unique experience to relate to and support these younger students and provide the type of support that I was in need of, but not receiving earlier in my career. Through working with students, I also started to see what Megan was talking about when she discussed superpowers. So among these students, I recognized unusual capacities for things like social perception, creative problem solving, kindness, empathy, and determination. And from my perspective, these are exactly the people who science needs. For example, with social perception, we can write better grant applications and make our science communications more relevant. Or with kindness, we can build better relationships with our peers and have healthier collaborations. These interactions started to show me that the problem is not with me for learning differently, but with an academic institution that has policies that prioritize learning and completing tasks in particular ways, and that these policies are disadvantaging students that learn and function differently. So I have 3 recommendations for changing policy and culture regarding disability within academia. So first, I recommend removing timed exams, which includes these standard entrance exams, as well as timed class exams. On timed exams, speed is used as a tool to evaluate who should be admitted to a university and how well a student understands the content from a class. Speed is not a measure of intelligence or of learning, and so I think it's really weird that speed is used in these contexts. Students who don't perform well on timed tests won't have the same opportunities to be admitted to certain academic institutions where they would otherwise perform well or to achieve high grades in their classes. Extended time accommodations are available, but the process of getting accommodations can be expensive, as it was in my case, and not all students will be qualified. Currently, under conditions of the COVID-19 pandemic, many universities have started dropping the entrance exams requirements, which I think proves that policy changes on this level are possible. But I think it's also important to pay attention to what's happening with this realm, because it's possible that these universities will revert back to these inequitable policies in the future once the health risks diminish. So for graduate students and professors, I encourage you to use learning evaluation techniques within your classes that don't involve timed tests, and to take action if entrance exams are starting to be reinstated. Next, I think that we should broaden access to learning accommodations. Current policies at OSU say that the only students who can receive formal learning accommodations for things like extended time or assistive technologies are those with diagnosed conditions that are recognized by the university. The policy excludes students who don't have a diagnosis, students who have a diagnosis that is not recognized by the university, or students who cannot afford a learning evaluation. The university should make classroom accommodations available to students who want them by removing the requirement for formal documentation of a condition. There's already a precedent for this type of policy change because because under graduate student employee contracts, they're allowed to seek work accommodations through equal opportunity and access offices, and they don't need to have formal medical documentation. So making learning accommodations more accessible allows more students to learn effectively and removes the unfair disadvantages that exist under current policies. And finally, I think that we should promote conversations around variations in need. We should not assume that everyone in a room has the same abilities or has the same needs. And we also shouldn't assume that in the classroom environment that all of the needs that exist are being taken care of by the disability access services. So if you create space for conversations around need, more people will feel safe to advocate for themselves and will be more likely to have their needs met. So for teachers, on your first day of class in your room, in first week in your classroom, you can ask your students if anyone needs anything to help them learn more effectively in this classroom or to complete their tasks at hand, please let me know. And so this acknowledges to your student that variations in need exist, tells students that you're ready to test their needs. It also allows students to advocate for themselves by answering questions rather than initiating the conversation. This technique is also relevant if you're facilitating a meeting or a group activity. And if you have any questions about any of the content that I've shared, you can email me. I'll have my email address up on the last slide. And so with that, I would like to thank everyone for coming out and participating in this presentation with me. This is a huge accomplishment for me. I would like to thank my lab groups, my advisor, the lab members, the undergrads in my lab. I have some specific people that mentored me at OSU outside of my lab group, the land managers and landowners that allowed me to access the field sites that I use, my family for always being on my team, my friends for always being up for fun and adventures, and then the mentors that I had from Bishop who helped prepare me for this experience and continue to support me throughout my career. And with that, I will take questions.
All right, I'll give the actual applause here since I'm probably the only one as a panelist who can do this. Thanks, Christy. And I should point out that the entire part 2 is also part of Christy's thesis. And so that's a perspective piece that we're hoping to prepare for publication as well. So let's see, I see we have a Q&A and we have a chat here. So let me see where the questions are popping up. So Teresa, can people actually ask their questions live? Is that reasonable to do under our Zoom format?
Yes,
So,
they just need to raise their hand and I'll activate them to speak and then they can demute.
okay, I see a question from Cheyenne in the Q&A. So does Cheyenne need to, does she need to raise her hand?
Yeah, she just did. She just
Okay,
did. So,
great.
Okay, she can now go ahead.
Can you hear me?
Yes.
Cool. Well, great talk. I really appreciated both the science and the perspective. It was really, really great. And thank you for sharing that. And I was curious, going back to your plot on, I think it was the non-insect predators during the filling period and the drying period.
Yeah.
And I think it was where in the drying period, the non-insect predators actually increased, which was like the inverse of what you were predicting. Is that correct?
The non-insect predators were higher in the filling phase and lower in the drying phase.
Okay, well then I'm going to ask the swap of my question then. Do you think that was because there was more nutrients available in the temporary ponds, or because it was like a concentrated source of prey?
So I think that a large amount of the variation that we observed was because copepods, which included calanoids and cyclopoids, are really high abundance invertebrate, and they're also predators, and they don't have protective body covering, so they're pretty vulnerable to predation themselves, even though they are predators. And so I think that that was a large influence on those proportions declining. And so it actually makes me think a little bit about these predictions under assembly that predation increases with habitat duration, but I think it might actually he's specifically referring to macro predators of like these bigger invertebrates or animals that are predators, and it's not necessarily considering that there actually is a high abundance of really, really tiny predators in the system. And so I think that it's a real trend, but I think that it gives evidence that these theories that are kind of shaping the way that we're making our predictions, there's some space for actually like improving and developing on those theories.
Sweet, thank you.
Okay, I also have a question from Scott. So Scott, you can go ahead and unmute yourself and ask your question.
Okay, can you hear me, Christy?
I can.
Great. By the way, I really appreciate the perspective on disabilities. For me as a teacher and mentor, that's really useful and helpful for me to hear that. So thanks for doing that.
Great.
So I'm curious, in your NMDS ordination, can you explain a couple of those outliers? Particularly, I think one of those is mine.
Yeah, it was yours. Oh, okay, let me go back to the screen share.
I don't know if you just have some guesses as to why those might be different.
Yeah, so my biggest guess is your pond is one of the deepest temporary ponds, and there is an association between pond depth and beetles colonizing the habitat. I think that beetles that are looking for permanent habitat are looking for deeper ponds as an indicator that the habitat is permanent. And your pond was really deep, and so I think that I'm actually going to share it so that we can see. Okay, can y'all see?
Yeah.
Okay, so yeah, so Scott's Pond is the one that's labeled as 147 and is over near the 365 plus in the top right corner. So that was the filling phase. So that sample had a lot of adult beetles in it, which I think were beetles that were flying in, migrating in aerially. And I think that they landed in the pond. This is my guess. I think that they landed in the pond, saw that it was deep, thought that it would be a permanent pond, so they tried to establish there. And then later on, when the pond started drying, it wasn't a good habitat. So I think that those same beetles likely emigrated back out. And I don't think that that happened with the other habitats in the area, because historically, in the Willamette Valley, the temporary waters were prairie wetlands, which were only a few inches deep. And so a pond that's like, I think that pond is like 5 feet deep. It was just about deep enough to top my waders. I think that that just kind of gave like an environment that was a little bit different from what the invertebrates were expecting. So that's my best guess. And then the other pond that was an outlier, that pond was full of filamentous algae, and none of the other ponds developed that, which I think was just an indicator of historic land use. That pond was in I thought it was a reclaimed agriculture area, but I'm now realizing that it's actually an active agriculture area. So I think that that's what affected the other outlying pond.
Great, thanks.
Mm-hmm.
I have a question in the Q&A and one in the chat. Would you like me to just read them out?
Yes, please.
Okay, so there's one in the chat from Rick, and it says, will your thesis present or reference files that give the abundance and identity of all the invertebrates in each pond?
Yeah, and I actually have a figure that shows that in my thesis, but I cut it out of my defense just because it took a long time to explain, and the big takeaway that I wanted to show was just that insect and non-insect communities were responding pretty differently over the course of the hydro period, and so I do have that information available. And I can share that if you send me an email, I can share that with you.
Perfect. And then I have one also in the Q&A from Ari Goodman. And they say, so do you think there's a path forward for institutions that value publication and productivity over individual or community needs? And what would that look like?
Wait, could you read that one again?
So do you think there's a path forward for institutions that value publication and productivity over individual or community needs? And what would that look like?
That's a good question, Ari. I would say that recognizing that that is an issue that's happening is a really big first step. And and recognizing that if that's happening and it's a problem, that it's a problem that should be solved. I think getting more people onto the team of understanding the problem with valuing productivity over individual and community needs. And I think that, in the suggestions that I gave in my final slide, I was trying to kind of cue students and professors to see the roles that they could play in making these changes. I think that community advocacy, so advocating for other people in your community that have particular needs that are not your needs, but you understand that these needs are important to someone else. I think that, kind of bridging these gaps to share, what the problems are and how these problems can be solved, and share them with people in our community who have influence and are interested in supporting us. I think that this is, the way that these challenges could be solved, because, thinking about our work a little bit differently of the quality of the work rather than the quantity of the work, I think could be a helpful way to kind of move forward.
And then there's also a question in the Q&A from Maggie Graham. And what is your favorite invertebrate that you sampled during this study, and why?
Thanks, Maggie. In a couple of my samples, I caught fairy shrimp, and fairy shrimp are my favorite because they look like alien fairies, and they're really cute, and they're super specialized to these temporary habitats. And it also, I think, is really special that they showed up in these habitats because the habitats I studied are all restored, so everything that's in them was carried in or moved in somehow, and fairy shrimp can't fly. So how did the fairy shrimp get there? So it just kind of brings us into like this bigger kind of like question of like what's happening out there, and like how are these little tiny critters moving around?
I don't see any more raised hands or questions right now. So
I see a lot of I love fairy shrimp emojis popping up in the chat though. So.
I know. I
Okay.
do. Jesse just asked another question though, Jesse Laney.
Okay.
He has two. He says 2 questions. What are your thoughts on the potential influence of vertebrate predators, amphibians/birds, on species composition and or diversity during hydroperiod in these ephemeral systems?
I think that that's a really good question, Jesse. So there were tadpoles in some ponds, not all ponds, and some ponds also had cattails that were creating habitat for the red-winged blackbirds. And so I do think that, and there were also geese in some sites. And so I do think that that was something that was influencing the invertebrate communities. However, when we looked at the ordination, it looked like things were similar. And there was a little bit of variation, on the axis 1 for hydroperiod length, which showed that, there was some variation based on hydroperiod length, but it wasn't super dramatic. And so I think that's a good question and potentially something to add into this type of study in the future. It was a bit bigger than what I had the scope for in my study, but could be a potentially good collaboration to have like potentially some amphibian and bird researchers, in collaboration with invertebrate researchers to kind of get a bigger picture of what the community is doing. And also these invertebrates could be providing like terrestrial subsidies in terms of like they when they emerge and fly out, they're eaten rather than they're being eaten in the pond. But I do think that these ponds are really important aspects of a productive ecosystem because there are so many vertebrates that use them as food sources.
Okay. And Jesse had another question, but I'm just going to email it to you really quick because I have one more person with their hand up as well. And so Ryan, you can go ahead whenever you're ready.
Hi, Christy. This is Ryan. Great presentation. Thanks so much for that. My question is, did you have any way, or did you try to characterize how wet or how dry the rainy season was that you were looking at this data? So like, was it, I could imagine if it was in particularly dry year, the hydroperiod for all the ponds would be shorter. Or if it was a particularly wet year, the hydroperiod for all the ponds would be longer. And did you characterize that in any way? And do you think that has an effect or would have an effect on the results?
Yeah, thanks for your question. That was something that we considered trying to look at early on when I was designing my project, is trying to look at satellite imagery to try to figure out historic hydroperiod lengths of the temporary ponds. And it ended up being pretty complicated because satellite imagery doesn't work with clouds, and it's always cloudy in the Willamette Valley in the winter. So that wasn't a great approach. But I do like actually what you were saying of like precipitation, of just looking at like, is it a wet year or a dry year, and using that as an indicator of variation. And so I think that was a limitation of my study, is that I only collected samples over 1 year. And so, I'm just sort of assuming that that's an average year, but it could be a particularly wet year or a dry year. And these things could have pretty big effects on hydroperiod lengths of the ponds. So I think that could be a good, development on this idea in the future.
Great, thank you.
That's it. That's all
Great.
I have.
Good, wonderful questions. Thank you, everybody, and thanks for a great talk, Christy. So more applause. Okay, well, let's we will end this session here, and I will ask the committee members to reconvene. Why don't we reconvene at 10 after, so at 10:10, and that's with a different link for a different Zoom meeting. So I'll look for that again and resend it out to everybody in case you don't have it. And we'll be there for the defense. Sound good?
Sounds good. Thank you.
Thank you, Christy. See you in a minute.
This chat's making me really happy.
Go ahead and bask in it.
Okay, I'm gonna shut us down.
Okay, sounds good.